Good evening, this is Dr. CB, PhD. It is Sunday evening, 10/4/2009, and today I'm going to be doing a video diary on microorganisms. So let me continue here. Viruses are genetic elements that can replicate independently of cell chromosomes, but not independently of cells themselves. In order to multiply, viruses must enlist a cell in which they can replicate. Such a cell is called a host. Viruses are characterized by also having a mature infectious form that typically extracellular. Viruses, like plasmids and some other genetic elements, take advantage of the metabolic machinery encoded by the host cell's own chromosomes. Like these other elements, viruses can confer important new properties on their host cell. These properties will be inherited when the host cell divides if each new cell also inherits the viral genome. These changes are often not harmful and may even be beneficial. However, viruses, unlike genetic elements, such as plasmids, have an extracellular form that enables them to be easily transmitted from one host to another. This extracellular form has enabled some viruses to replicate themselves in a host in a way that is destructive to the host cell. This destructive replication accounts for the fact that some viruses are agents of disease. In many cases, whether a virus causes disease or hereditary change depends on the host cell and on the environmental conditions. Because viruses have a form of independent of cells, some people refer to them as living organisms or life forms. As we will discuss below, however, some properties that serve as characteristics of living systems do not occur in the extracellular form of the virus. Without host cells in which to replicate, there can be no viruses. In this chapter, we shall discuss some of the ways in which viruses can redirect the metabolism of the host cell in order to replicate. This chapter is divided into 4 parts. The first part introduces basic concepts of virus structure. The second part deals with the host cell and how viruses can be quantitated. The third part deals with some of the basic molecular biology of virus multiplication. The fourth part gives an overview of a few of the viruses that infect bacteria and animals. Further material on specific viruses can be found in Chapter 16. These discussions expand on the concepts of macromolecular synthesis, of gene regulation we covered in Chapters 7 and 8. Viruses are among the most numerous microorganisms on our planet and infect all types of cellular organisms. Therefore, they are interesting to study in their own right. However, scientists have also studied and continue to study viruses for what they can tell us about the genetics and biochemistry of cellular metabolism. And in the case of some viruses, the development of disease. Furthermore, as we shall see, viruses are also important tools for microbiology geneticists and the genetic engineer. Virus and virion. General properties of a virus. Here we describe both extracellular and intracellular states of viruses. In the extracellular state, a virus is a minute particle containing nucleic acid surrounded by protein and occasionally, depending on the specific virus, containing other macromolecular components. In this typical extracellular form, the virus particle, also called the virion, is metabolically inert and does not carry out respiratory or biosynthetic functions. The virion is a structure by which the virus genome is carried from the cell in which it has been produced to another cell where the viral nucleic acid can be introduced. Once in the new cell, the intracellular state is initiated. In the intracellular state, virus replication occurs. New copies of the virus genome are produced, and the components that make up the virus coat are synthesized. When a virus genome is introduced into a host cell and reproduces, the process is called infection. A cell that a virus can infect and in which it can replicate is called a host. Viral genome are limited in size, and they encode primarily those functions that they cannot adapt from their host. Therefore, during replication inside a cell, viruses depend heavily on host cell structure and metabolic components. The virus redirects pre-existing host machinery and metabolic functions to support virus replication and the assembly of new virions. Therefore, most virions can also be found inside the host late in infection. Found inside the cell late in infection. Viral genomes. All cells have double-stranded deoxyribonucleic acid, DNA, as their genetic material. In contrast, viruses can have either DNA or ribonucleic acid, RNA, as their genetic material, and it can be either single-stranded or double-stranded. Viruses are sometimes divided into two types based on whether they have DNA or RNA, as their genetic material, and all viruses contain either one or the other, and virion. However, there is a third group of viruses that use both DNA and RNA as their genetic material, but at different stages of their reproductive cycle. This last group includes the retrovirus, which contain an RNA genome in the virion but replicate through a DNA intermediate, and the human hepatitis virus, which contains DNA in the virion but has an RNA intermediate in replication. These classes can be further subdivided according to whether the nucleic acid in the virion is single or double-stranded. The classification of virus based on the types of nucleic acid in the virion and the associated replication strategies have been formalized as the Baltimore classification system, which we will discuss. Despite the diversity of genome structure, viruses obey the central dogma of molecular biology. All genetic information flows from nucleic acid to protein. In addition, all viruses use the cell translational machinery. And so, no matter what the genome structure of the virus messenger RNA, mRNA must be generated that can be translated on the host ribosomes. Viral host and taxonomy. Viruses can also be classified on the basis of the host they infect. Thus, we have animal viruses, plant viruses, and bacterial viruses. Bacterial viruses, sometimes called bacteriophages, or phage for short, from the Greek phagine, meaning to eat, have studied primarily as a convenient model for systems for research on the molecular biology and genetics of virus replication. Many of the basic concepts of virology were first worked out with bacterioviruses and subsequently applied to viruses of higher organisms. Because of their frequent medical importance, animal viruses also have been extensively studied. The two groups of animal viruses most studied are those infecting insects and those infecting warm-blooded animals. Plant viruses are important in agriculture but have been less studied than animal viruses. Finally, there is also a formal system of viral taxonomy that organizes viruses with the hierarchical taxon levels, order, family, and subfamily genes, and species. Although at times such formal taxonomic system can seem quite arbitrary, and common names are still wide use, the system is increasingly useful because of the increased phylogenetic data being accumulated. For family taxon seems particularly useful. Members of a family of viruses have distinct virion morphology, genome structure, and or strategies of replication. Virus families have name including the suffix viridia, and in poxviridia, and we will discuss a few of these in Chapter 16. Nature of the virion. Virus particles, virions, vary widely in size and shape. Viruses are smaller than cells, ranging in size from 0.02 to 0.3 microns. A common unit of measure for viruses is a nanometer. which is 1,000 times smaller than 1 micron and 1,000,000 times smaller than 1 millimeter. Smallpox virus, one of the largest viruses, is about 200 nanometers in diameter, a bit smaller than the size of the smallest bacteria. Poliovirus, one of the smallest, is only 28 nanometers in diameter, about the size of the ribosome. As we have stated, some viruses contain RNA, others DNA, and the nucleic acid can be either double or single-stranded, depending on the virus. Viral genomes are also smaller than those of the small cells. Most bacteria genomes are between 1,000 to 5,000 kilobase pairs of DNA, with the smallest known being 580 kilobase pairs. Interestingly, bacteria with the smallest genomes are like viruses, parasites that replicate in other cells. However, the largest known viral genome, that of bacteriophage, G, is 670 kilobase pairs. The virus which infects Bacillus megaterium is, however, one of the only of a few viruses currently known whose genome is larger than cellular genome. More typical viruses' genome sizes are listed. Some viruses have genomes so small they contain less than 5 genes. As can be seen in the table, the genome of some viruses, such as the reovirus, is segmented in more than one molecule. The structures of virions are quite diverse, varying widely in size, shape, and chemical composition. The nucleic acid of the virion is also always located within the particle, surrounding by a protein coat called the capsid. The terms coat, shell, and capsid are often used interchangeably to refer to this outer layer. The protein coat is always formed of the number of individual protein molecules, also called the structural subunits, which are arranged in a precise and highly repetitive pattern around the nucleic acid. The small genome size of most viruses restricts the number of different viral proteins. A few viruses have only a few kinds of protein in their capsid, but most viruses have several chemical distinct structural subunits that are themselves associated specific ways to form larger assemblies called capsomers. The capsomer is a morphological unit that can be seen with the electron microscope. The information for proper aggregation of the structural subunits into capsomers is contained within the structure of the proteins themselves. And the overall process is assembly and thus self-assembly. For many viruses, this self-assembly process is assisted by molecular chaperones, proteins that assist in folding and assembly, but that themselves are not a part of the final structure. A single virion generally has a large number of morphological units, the complete complex of nucleic acid and protein packaged in the virus. Particle is called the virus nucleocapsid. Inside the virion are often one or more viruses, specific enzymes. Such enzymes usually play a role during infection and replication during, as we will discuss later in this chapter. Although the virus structure just describes is frequently the total structure of virus particle, a number of viruses have more complex structures than enveloped viruses. The nucleocapsid is enclosed in membrane. Viruses without membranes are sometimes called naked viruses. Virus membranes are generally lipid biolayer membranes, but associated with these membranes are often virus-specific